For the reproductive system, we start with the male reproductive system, and we will take a look at the difference between sexual and asexual reproduction. We will also look at some of the developmental processes from the female and the male, and we will go through those developmental stages and then even speak some on sexual response. So the anatomy, all the organs associated with each the male and the female reproductive systems, and the physiology, and then look at some pathophysiology as well. And this will conclude anatomy and physiology to your study of from this. So let's go ahead and jump right in here with the male reproductive system. So when we think about sexual reproduction, this means that it's going to require two parents. There are many species that actually use asexual reproduction, which means that one organism has the ability to just divide and make, the next offspring, and well, they're not really considered the next offspring, but the next generation without another partner. So all the genetics comes from one parent. But in the animal kingdom, most are sexually reproducing now and have evolved to be that way. So two parents. What determines the gender or the sex is going to be the male parent, because the male parent on the 23rd pair of chromosomes has an X and a Y, but the male can only send either the X with the sperm or the Y, so can only send one or the other. A female at that 23rd site has two X's, and that's all a female has. So they can send one of their X's to an egg, or they can send the other one, but a female only has an X chromosome to give to an egg, where the male depends on which sperm makes it to the egg. Is it the sperm that I got the Y, meaning that that new developing organism will be a male, or is it the sperm that had an X from the father, and then that X would join with the mother's X in the egg, and then that would be a female. So that's what we're going to take a look at. There will be some terms that will be repeated from our study of genetics. So I'm sure you remember that was way back in AMP1, near the beginning of AMP1. So some of the terminology you may have not heard for a while. You may have to refresh yourself on it, but you would want to do that. So two parents, a mother, a male and a female. I guess I shouldn't say mother and father. It's a little bit of a different connotation, but definitely male and female, where the gametes are called sex cells. So gametes are sex cells. The male gametes are sperm. The female gametes are eggs or ova. And we understand that gametes, if you hear the term gametes, it just means the sex cells. The very first cell that's formed when a sperm meets an egg at fertilization, the very first cell that's formed is called a zygote. So again, I think most people kind of understand that, and that it is the male parent that determines the gender of the offspring. When we think about the organs that are a part of the reproductive system, the primary sex organs are the gonads. So the gonads in the male are going to be testes, and the gonads in the females are going to be ovaries. Secondary sex organs also are vital. They're essential to be functioning, because if not, a female could be producing all kinds of eggs, but if the structures that support them and are needed to be supported along the way don't work, then fertility is never going to happen. Same thing in the males. The male may be producing sperm, but if there's a problem with the duct work, essentially it's the duct work, then the sperm may not be getting to go to where it needs to be. So all of the secondary structures need to be functioning as well. The ducts, the glands that are secreting their products, the penis that actually is used for delivering the semen, that has the sperm cells in it. Semen, ejaculatory fluid, about 10% of that fluid typically is actual sperm. The rest of the fluid is supportive and lubricating to support that sperm as it's being delivered. to the female. The female uterine, in a female, the uterine tubes, which are sometimes referred to as fallopian tubes, the uterus itself, the vagina, that receives the sperm and then acts as a birth canal when the fetus is being delivered. So these secondary sex organs also must be healthy and they must be functioning. It isn't just about the testes and ovaries. The secondary sexual characteristics also are important, and when we think about those, we think about the things that you see, the changes that are on gross anatomy that happen at the onset of puberty. So in males and females, there's the pubic and axillary hair. With males, the testosterone affects the larynx, so the voice starts to change and deepen and take on a different resonance. Beard growth in males, we know. Scent glands that are secreting pheromones to even subconsciously the opposite sex are picking up on those pheromones to know that there's a preparedness and maturity that is happening for reproduction. You know, I think sometimes humans don't like to think of ourselves as animals, but we are animals, and all of these secondary sexual characteristics play a role subconsciously and even consciously in attracting a mate of the opposite sex. For, you know, because it's about procreation, it's about reproduction, it's about the next generations and keeping life going. There is a typo here on this slide, and I tried to change it, but it won't let me. Eggs only have X's, and they only have one of the X from the mom. So when we know that cells start off with 46 total chromosomes, but 23 pairs, but when a female is making eggs, the gametes, only one from each of the pairs can go, only one homolog chromosome from each of the pairs can go to the egg. So again, on the 23rd site, it might be one X or it could be the other X. Females don't have the Y. But the sperm will either be holding an X in the 23rd pair or a Y, and it's usually 50-50. So males are, when they're producing sperm, about half of the sperm per ejaculation are going to be X and the other half may be Y. Okay, so that's just sort of a visual on that. So when we are thinking about embryonic development, I don't ask any questions about this on tests or quizzes, lecture quizzes, nor tests, because embryology is almost like a study all in itself. But I don't ask questions about the differences between 5 and 6 weeks, and then to 8 and 9, and then at 12 weeks into birth. There are a lot of different developmental stages, but I do want you to know that on that Y chromosome, there is a region called the sex-determining region of the Y. That region is actually going to be where the genetic information is held that will turn on testosterone during embryonic development, and that is what causes the tissues to end up, that organism becoming a male. At 5 to 6 weeks, there is really no difference in a developing embryo. There is no difference. You know that from 2 to 8 weeks, this developing organism is called an embryo. From 9 weeks until birth, it's known as a fetus. But during the embryonic stage of 5 to 6 weeks, there is no significant difference. That SRY gene that's only found on the Y chromosome will be active for just a very short window of time, a few hours near this time. And what it does is it causes a flooding of the testosterone and that Mueller inhibiting factor that is then going to start changing and affecting some of the tissues that are present here, and that testosterone will dictate that these structures become male. So the testes are beginning to develop, and eventually the testes will drop down out of the abdominal cavity, the pelvic cavity, and drop down into the scrotal sac. But here they're forming at 7 to 8 weeks, and by birth the testes have actually moved through a structure called the inguinal canal on the right and the left side, and now the testes is in the scrotal sac. the tissues that if testosterone and that Mullern inhibiting factor is not present, and the absence of the hormone flooding the organism, those structures will continue to develop into a female. So you can see very much the sort of symbiotic relationship of some of these structures and then how they're developing. The ovaries remain in the pelvic cavity along with all the ductwork for females. The structure that becomes the penis in a male will become the clitoris in a female. The structures that become the testes in the male, the primary organs, become the ovaries in a female. The structures that become the scrotal sac in the scrotal sacs here in a male, the same tissues, become the labial folds, the external genitalia of a female, the labia, the lips, the outer lips they call them. So again, males will form in the presence of that testicle because of the SRY gene, which only works for a few hours during embryonic development, will cause these tissues to become male. In the absence, a female is going to be at birth. So the development of the external genitalia, you can kind of see, it would be very difficult to tell the difference between the glands of a penis and a clitoris. But then as the fetus is developing, you can see that you start to see some major differences here. The testes themselves start off in the pelvic cavity, but they're going to have to descend into the scrotal sac. There are about 3%, which is fairly significant, are born with their testes that have not descended. So the scrotal sacs on examination are empty. And so, this is one of the things that you look for in male babies, is that the testes are actually, you can palpate and gently and palpate the testes and see that they are in the scrotal sac and they have descended. Sometimes they float, so they will sometimes be there, and then you'll have parents bring their male babies in and they're very panicked because they have noticed that the right or the left scrotal sac is flattened and empty. And you can imagine how alarming that would be for a parent who didn't understand that sometimes those testes do float, but they should descend and stay descended within a certain period of time. They should be descended at birth, but they should certainly be staying descended within a certain period of time. And they really watch for that, because if this testes somehow gets trapped in the inguinal canal with the spermatic cord tightening it down, or if it hasn't descended and it's still in the pelvic cavity, it will die after a certain amount of time. So certainly pediatricians look for that. They sometimes talk to the parents about that if they think that there's any issue with that, and they can go in, and if that is the case, they can go in and pull that testicle down along with all these accessory structures, and they can do a little tie off themselves. They do have to be careful, because you can imagine it's not just the testes that descends into that scrotal sac. It's all of the ductwork, the epididymis, the ductus deferens, and I'll talk about these structures in a minute. But it's really the ductwork that has to come down too, as well as nerve supply and blood supply. So you certainly wouldn't want to strangle this area. So they're, pediatric surgeons actually specialize in doing these particular procedures if they need to do that. The boundary that, so I did want to tell you, I don't know if I mentioned the word or pronounced it for you, but it's called cryptorchidism. It is the term used for undescended testes. So cryptorchidism is that term that is actually used. The male perineum, we think about this particular, structure, this shape is containing this. We don't often refer to like the male perineum, but you'll see similarities of these terms when we do talk about the female perineum, because the female perineum, for pelvic exams and whatnot, we do look at. This mid-sagittal section, so everybody remembers that a mid-sagittal section separates the right from the left. So what we're looking at is an illustration of the mid-sagittal section of the male reproductive system. And we see kind of right away that, and we talked about this in the urinary system, the male reproductive system shares structures with the male's urinary system. So if we think about sperm forming in the primary sex organ, which is the testy, that sperm is going to have to move along this ductwork. This is just the right one. There's also a left one. So move along the ductwork all the way back up into the pelvic cavity, and then it joins at this ejaculatory duct with the left one, because there had been the left side as well. They join here at the ejaculatory duct right at the prostate gland. The prostate gland completely surrounds this prostatic urethra and this ejaculatory ducts. It's a gland that's about the size of a walnut. Right here, it looks ivory on this picture here. This is the whole prostate gland. And this ejaculatory duct is where semen is deposited from the right side and the left side. And then it will move through that prostatic urethra to the penile urethra, the penis urethra, until it's out. So we do know that there are shared structures. When ejaculations occur in a male, there's a valve that closes off the urinary bladder. So urine does not mix with semen. Urine does not mix with semen. So there you will also notice that the prostate is a gland, so it is secreting prostatic fluid that contributes to about 30% of semen is from the prostate gland, that fluid that is nourishing and also protecting the sperm. So does the bulbourethral glands, so you can see this here, and the seminal vesicles. These are all glands that are secreting, contributing to semen. And as we said, only about normally, depending on a lot of factors like hydration and, just a ton of factors, only about 10% of an ejaculation is actual sperm, but the rest of it is absolutely necessary. It's necessary for propelling along, mixing, protecting, and nourishing the sperm that's there. So all of the fluid that we think of is important for that. This mid-sagittal section picture, I could pretty much do the entire lecture kind of talking about this just from this picture, and I'll come back to it. It looks like so much is actually labeled, and it might look a little bit intimidating, but this, it shouldn't though, because we're going to end up talking about all of these structures, and you should be sort of visualizing them, because part of your lab assignment will be that you do your own kind of drawings here and are able to identify these structures and know them in the male. So let's take a look at what we already know. We know about the urinary bladder in the male. So this bladder is a muscular sac that actually will fill and receive urine from the ureters, from the kidneys, and be able to hold urine between voids. So then there is this internal and external sphincter that the male can control when urinating after about around the age of 2. But this sac is a muscular sac that will contract when voiding urine. We know about the pubic region of the pelvic bone, called the os coxae, is the pelvic bone, and this is the pubic region of that, called the pubic symphysis, where the right pubic region of the os coxae joins or meets with the left pubic region of the left os coxae. So that's where this is, what this is. We know that as far as the penis goes, that what people call the head of the penis is actually called the glans. Please look at the spelling here. This is different from a gland that secretes. Glans is the very nerve sensitive, very sensitive end of the penis, referred to as the glans. And then the urethral opening the urethral opening, should be at the tip of the glands. Sometimes it's not. Sometimes this opening is actually going to be in a, it can be underneath, it can be whatever. This is referred to as hypospadias. So this is a term that you'll see in your disorders table, and they can repair that. So they can surgically correct that after a certain age. They sometimes don't do it right away, depending on some factors from the neonate, but they can repair that. There is tissue that actually protects the glands. It is so sensitive that there is a tissue that protects. It's called the prepuce. People call the prepuce foreskin in males. They call that foreskin. And sometimes foreskins are electively removed. I mean, people choose to have those removed. There is no medical reason to have foreskins removed. There has been lots of studies and lots of retrospective studies, lots of ongoing studies about this, but there is no real medical reason for foreskins to be removed. The reason, when we look back historically, we see that this is a practice that has been done sometimes for religious reasons, sometimes like in some of the major religion, there have been thousands of religions, and there still are a lot of religions, but some religions do it. It's very documented in their literature that this is done as sort of a proof for serving of a higher being. Some cultures or religions do it as a rite of passage into adulthood, so like a puberty type of rite of passage. So we've seen a lot of different reasons that sometimes this practice is done, where the foreskin will be, the prepuce will be removed. But again, there is no medical reason for it. So in most developed countries now, today, insurances aren't going to always be covering this anymore. So now we see, and it's sort of a pendulum swinging back and forth, but now we see, when I say pendulums, as far as the practice, like some will go through periods where there are a lot of people, most people are getting males are getting circumcised, and then we go through periods where it's not as many getting circumcised as not. And so, sometimes that pendulum of that cultural practice will swing, is what I'm talking about. I'm not talking about anything anatomically swinging. Y'all will forgive me for that, but I was afraid somebody might be thinking that. So anyway, right now in this country, there are less males being circumcised than there are not being circumcised. So right now, the circumcised male babies are in the minority, where for a long time in this country, that wasn't the case. So, again, go through different eras of that as far as what people are doing. But it had been thought at one time, people thought it was a cleanliness thing, or a reason that males would have less urinary tract infections if they were circumcised. But that has not been proven the case. Of course, genital hygiene is important in both males and females, so both males and females. Females, the tissue that is a penis in a male, you can see they've illustrated a type of tissue here. It's called erectile tissue. These erectile tissues. These tissues in a female become the clitoris. And the clitoris has a prepuce too. Some people refer to this as the clitoral hood. So again, in an embryo, if during that little short window of time that the SRY gene is active and flooding with testosterone, the tissues will become, that are exposed to that, will become a penis. In the absence, they become a clitoris, and the prepuce over that's protecting the clitoris it's called a prepuce. So anyway, there's risk to this surgical procedure that is done in male circumcision. There's always a risk, and there's a certain percentage of complications that happen. So this is not a procedure that's done that has no risk. There definitely things can go wrong, in this. And again, there's just no real medical reason for having it happen. In this picture, you can actually see the testy here, and we'll see a little cross section of it. And when we look at the testicle here, we can see, I think I had a picture, I don't know. Could have sworn I had a picture of that. Okay, so this is kind of a dissection here where you see all these little ductworks inside. So this testy has been cut, this right one has been cut. So you can see all of this. I think I have a better one as well. But anyway, so when we're looking at the testes, we know that this is where sperm is actually generated. So in these seminiferous tubules, these little microscopic tubules, the sperm is actually produced. The sustenticular cells are actually going to help with the development. These are going to be cells that we do know are going to help to provide a blood-testes barrier. When sperm is actually forming and those sperm cells are created, they're no longer really the male's typical cells with 46 chromosomes. So if there wasn't a barrier, the male's immune system wouldn't want to actually attack the sperm cells. There are some males that are infertile because this blood-testes barrier has actually been breached and there's no longer protection. So the male's immune system ends up attacking their own sperm. So some males, this is a rare thing, but some males are actually infertile for that reason, or have a very much of a lower sperm count or viable living sperm count. So it's much more, of a problem than that. So this is where sperm are formed, and when the sperm are maturing, they're moving through these little ducts. It's like a highway, and they move through these little ducts to this area called the epididymis. Sometimes males have epididymitis. So that's inflammation of this particular structure. But anyway, this is where the sperm are going to finish off their maturation process. So they're going to finish off maturing here, and after they've reached a certain maturity, they're actually going to move along this ductwork all the way through to the ductus deferens. Now I'm going to go back. This was the epididymis, where they're sort of finishing their maturation process, if you will. They're going to school. So this is like their school. But once they're mature and they finish it off, they're ready to be delivered. So the duct that is going to propel them back up through, it's a long way to go, propel them back through to the ejaculatory duct, through the prostatic urethra, through the penile urethra, and out. So that's the journey that they take. So as I said, this looked like a lot in the beginning, but now we're talking about most of these structures, so I hope it doesn't look so overwhelming to you now. And again, most of the lecture really kind of being done from this particular illustration, just because it makes it easier to see. I want to let you know that men can actually have an in-office. Of course it is invasive, but it's sort of a minor procedure that, easy for me to say, right? Minor procedure for men to have what's called a vasectomy. A vasectomy is done in a urologist office. They will palpate the scrotum until they can feel this, and they really are just palpating it until they feel this ductus deferens. They can, manipulate it pretty close to the surface of the scrotal sac, make a very superficial incision, and cut this duct, and then band the ends off. Tie or band. There's different things that, depending on the urologist, procedures they would do, and that's called a vasectomy. The reason it's called a vasectomy is that this duct that you see is labeled as the ductus deferens is also more commonly referred to as the one word vas, V-A-S, vas deferens, and a vasectomy means the cutting of that. And of course they would do the right and then they would do the left. It takes not very long. It takes very little bit of time to do that. They do local numbing, so the anesthesia part is not dangerous. The procedure is not that invasive as compared to if a female has her tubes tied, the fallopian tubes, uterine tubes, that's major surgery with anesthesia, and then the major surgery, the complications that can go along with major surgery, because they have to get into the pelvic cavity. With this, not as serious. So anyway, the problem comes in with vasectomies in that sometimes when these are done, there's still sperm that are along these tubes. So sperm can actually be in this tube and still be part of the ejaculatory process with seminal when they're contracting, when these seminal vesicles and prostate gland and bulbourethral glands are secreting with that ejaculatory process. These sperms can live here for weeks. So I think the recommendation is like at least 6 weeks to still have protected contraception, because for 6 weeks there could have possibly been sperm that would have been in this tubule that would, could actually cause a pregnancy. And then they don't tell you to stop using an alternate way of contraception until the male comes back in and they give a semen analysis and they see that there are no viable sperm left in the ejaculation. So it's important that they follow those steps before they think that they are using a vasectomy as a form of birth control. There have been probably more than there should have been pregnancies that happened just because people thought, oh, it'll be kind of safe because I had that vasectomy a week ago or even a month ago. That you need to be careful with that. But anyway, so that's what they're talking about with the vasectomy. I'm trying to think if there's anything else on this picture. You can see from the rectum here, and the rectum, and then the anal orifice here, you can see why a rectal exam for a prostate, to really palpate that prostate, is actually a good way to for a urologist to be able to actually palpate this prostate to see if there's anything going on. Another thing I'll tell you, since I have you on this picture and you can see, men as they age, it's normal for their prostate gland to enlarge, and that's a normal thing that we know happens with males' prostate glands. And as those prostate glands enlarge, and then you've got all the neighboring tissues, it can kind of close off. Not completely, it doesn't close it off, but it can actually cause the urinating process to be impeded. So men can have, because of an enlarged prostate gland, they can have trouble like completely emptying their bladder. So it can actually cause some frequency in urinating, so then they have to urinate more frequently, or they're up at night, urinating more frequently. That can be just really normal, and it can be hard for them to completely empty. It can also cause some incontinence issues. So this can be something that men as they're aging can face. Another thing, if men live long enough, if they live long enough, and we know life expectancies are, have increased, almost doubled in just the last short period of history for man, in the last couple hundred years, our life expectancies have doubled. If men live long enough, prostate cancer is pretty common. But there are different types of prostate cancer. Some prostate cancers are pretty aggressive, but most are not. So if males have the type of prostate cancer that is not aggressive, that has been detected when they're in their 70s, sometimes even 80s, they should just leave it alone. Really. Because going in and doing anything, there are so many accessory structures here, it can really cause incontinence issues, and anything that they're doing, whether it's radiation or it's certainly removal. This is a very serious, when they remove a prostate gland, this is a bloody kind of surgery. So oftentimes patients lose blood, and it can affect nerve supply to this region, the genitalia of a male. It can affect sexual activity. It can affect urinating. So it's a big deal to have a prostate removed. Radiation can be a big deal too, unless they're doing proton radiation. But anyway, if it's really slow growing, it is going to take 25 years to actually cause problems. So, they usually just leave it alone. Now, if it's prostate cancer that's in a younger person, 40s, 50s, even 60s, y'all are thinking 60s not young, but actually that can be kind of young. They're going to biopsy, they're going to test it, and if it's an aggressive type of prostate cancer, they have to treat it. Then they have to treat it, especially in younger men. So I hope that made sense. Now, there's also testicular cancer. So when males hit puberty, they should be taught that if there's any changes in the scrotal region, if there's any nodules that, they can palpate that they don't think are normal, and I'm not talking about the testes as a nodule, I'm talking about abnormal structures. If there's any soreness or tenderness, they need to check, because there's also testicular cancer. And testicular cancer we often see in young males, so teenage, 20s, 30s, so young. So men should be doing self-exams the same way that young women are taught to do self-breast examination. Men should be doing the same thing. Okay, I think I've really talked a lot about what I like to talk about in the male reproductive anatomy. This is an anterior view, obviously from front on, in a superficial dissection. So just getting down to the fascia, you can see all these important structures that have moved through the inguinal canal of the male, and how important that those structures are for the nourishment of the testes and all of the ductwork that goes along with the male reproductive system. Sometimes men will have hernias at this particular region, so part of their intestines will slip down into this region, and that can actually strangulate, cut off blood supply to not only the intestines that are being strangulated, but also some of these structures, important structures here of the reproductive system. So you can see the prepuce here, the foreskin, an illustration of that, where when the prepuce, just for basic hygiene, the prepuce can be pulled back and cleaned and whatnot, but it protects, it's very protective of the glands, which is, highly sensitive. The scrotum, they talk about the scrotum as being a pendulous pouch, and so I think that's what I was thinking about when I used the term pendulum. I just meant how sometimes cultural things and practices will shift, and then the time, then they become not less popular. So I wasn't talking about actual anatomy, but here they're talking about that. Okay. The scrotum is important though, because not only is it protective and housing the testes, and it's important that the testes descend out of the pelvis, because I said that they would die if they don't, right? It is much warmer in the pelvic cavity, much more warmer, and sperm couldn't develop there anyway. Sperm can only develop in cooler temperatures. They cannot develop in the core temperatures of the body, so we absolutely need them to be there for that reason. And the cremaster muscle of the scrotum and the dartus muscles you're seeing, these are going to be the muscles that actually lift the testes in times when it's really cold, and or allow the testes to be further from the body when it's really warm or hot outside. Again, these are very important that these are working as part of the autonomic nervous system. Men don't have to think about the temperature of their testes. This is the autonomic nervous system keeping sort of a check on the temperature in this region. So that's when it's really cold, the testes get pulled up. And I'm not telling you all anything you don't know, but that's because they are really incubating those sperm that must be between a particular temperature to be developing as they should. So there have been some men that have come in with fertility issues that they realize, oh, their clothing are too tight. So or that they are hot tubbing too often, because they talk to them and they say, well, it looks like you have sperm, but they don't look like they're alive. And because when they do a semen analysis, they also judge the viability and the motility of the sperm. They want to see the sperm moving at a certain pace and that there are enough of them that are actually viable, alive. And so sometimes men come in and then you start asking questions like, okay, well, there are a lot of sperm, but they don't look like they're active and they don't look like they're living. So can you tell me, you start asking questions like, what kind of clothing do you wear? Are you taking hot baths, or are you hot tubbing too often? And sometimes it's just as simple as changing your garments or changing some of your practice, as for males, changing some of the practice to actually get the sperm viability numbers up. That would be an easy fix, wouldn't it? So anyway, this again talks about peristaltic contractions during orgasm. So that ductus deferens, where the sperm have to travel through, there are coordinated muscular contractions propelling those sperms along. And you can see it's a significantly long tubule for it to get there. And the ejaculatory duct itself, again, you're looking at an anterior view of just some of these important structures. You see the seminal vesicle, which secretes fluid for semen, the prostate gland secreting fluid, the bulbourethral gland, both sides secreting fluid. So just another view here for you for that. And again, the picture, that all-important picture, lots of times you see it. Look at all of these just to see, and look at others if you want to. A transverse section so the penis actually being cut, like a cut at this point. A transverse section of this, you're actually seeing some of those tissues that we think of that are going to be necessary for the blood to actually move into the penis, to engorge the penis for what is known as an erection. So, so that's also important that happens. I don't think there was anything. Again, the urethra needs to open at the tip of the penis and not any other places. At puberty, we know at puberty there's this onset of those gonadotropins, FSH and LH in males. Go back and review that from the endocrine system. FSH is going to target the testes for sperm production. LH is going to target the testes for testosterone production, which is also going to play a role in finishing off that sperm maturation, and also all of those secondary sexual characteristics we think of with male. The deepening of the voice, the body hair that you associate with reproductive maturity, muscle mass increases in males. Males have a much higher muscle mass than females, just normally, physiologically. So that is going to also mean that they need to have a higher hematocrit because the muscles are so dependent on oxygen, and red blood cells deliver oxygen. So all of those things are happening. We think of puberty as being when, on a male, when the first viable sperm are present in an ejaculation. Males are ejaculating way before then, but when there's first viable sperm, living sperm, is sort of that area that we say is, sexual maturity. That doesn't mean that everybody should be having babies then. I'm just saying that physiologically, anatomically, they're capable, okay? The first menstrual period of a female is where they're said to have hit puberty. It will be a couple of years before this that some of the characteristics, the changes are starting to occur. But with the first menstrual period, then it means the female is going to be able to actually get pregnant. So adolescence in medicine, it's a weird definition in medicine, but in medicine it is defined as when a person finally reaches their full height potential. Females do that very early, and males sometimes they're in their early 20s before they reach their full height potential. Some males takes that long. So I'm not sure about this, definition for adolescence being just linked to continuing to grow in height, but that is what we do in medicine. So again, effects of testosterone. It's so many effects, and we can't forget that it's also the effect for libido, which is sex drive. So men have testosterone not just from the testes, but also from their adrenal glands. And men are producing sperm on essentially a 24-hour cycle. So, males really do have a very high, much more of a higher libido, sex drive than females do. We'll see when we get to the female system that females are really only reproductively capable with a viable egg, one egg, in a short window of time in a monthly cycle. So the sex drive, really the amount of testosterone in most females is significantly lower than in individual males. So, okay, again, this is a classic kind of negative feedback that occurs for these hormones, and none of these hormones should be new to you. Males can sometimes have too many sperm. And if they have too many sperm, oftentimes the sperm are there, but they're not viable, they're not active, they're kind of slow. So there needs to be just the right amount of sperm per cc or ml of semen. And what controls it is inhibin. We had talked about inhibin. It is going to turn off FSH, and when it turns off FSH, it's going to control the sperm count. So keeping the sperm count right where it needs to be, okay? So I think that these are all, you've seen these hormones before. As far as development and how these hormones actually are secreted, there's a decline in testosterone secretion, after the 20s. So it declines to about a fifth by the age of 80. But the fact is, males, very much unlike females, males can be fertile their entire lives, even if they live to be over 100. They would still be making, unless something happened to them, they would still be making sperm. And again, where females have a 28 average day cycle, it's average 28 days. Some females a little longer, some a little shorter, but the average female, 28 days. Males have a 24-hour cycle of making sperm, and they have the same kind of hormone fluctuations in a 24-hour period than females have in a longer spread out period. So the rise of FSH and LH after the age of 50 actually puts men in what is referred to as a male menopause. Climacteric is referring to menopause. So males actually go through that as well. So sometimes males in this age range, late 40s or 50s, they will have the same kind of mood changes, hot flashes, feelings like they're suffocating. Sometimes they come into ERs and they really are thinking maybe they're having a heart attack or an anxiety attack or something like that, but it's actually just the fluctuations of these. And typically they kind of resolve, just like with female menopause. The difference being that after the female's menopause, the female's no longer producing eggs, where again, males continue to produce sperm unless something else has happened that causes it to not. Naturally they do, for their entire life after puberty. Impotence, where there's an erectile problem, impotence, erectile dysfunction, happens. It can happen, but that has nothing to do with really ejaculation and viable sperm. So it has to do with like intercourse, because an erection is required for that, but it doesn't have anything really to do with fertility. So when we think about the production of eggs and sperm, there's a very special type of cell division that must happen. That cell division is called meiosis. So meiosis is only happening in the gonads. It only happens to get to the gametes. It's very important that you understand that. Mitosis, we talked about for cell regeneration, cell growth. You start off with one cell, the zygote. That cell divides. Those two cells divide, continue to divide. Throughout your life, you're repairing, regeneration is happening, all that's happening. That's mitosis. You start with one cell, you go through a cell division, and now you've got two daughter cells that are identical to the first parent cell. That's mitosis. That's not what's happening in the gonads. In the testes, you start with one cell, you go through two divisions, and you end with 4 gametes that have half the numbers of chromosomes. So I'm going to show you this here. Now in this picture, it's important to understand that you're seeing pairs of chromosomes. You understand that there are, in this male, there are actually 23 of these pairs. There's not just 2 pairs in a male. There's 23 of these pairs because there's 46 total chromosomes. Each of the partnered pairs are going to actually have this copy here, and in the first division, those numbers are going to be cut in half. So now you end up, after the first division, you end up with 2 cells that have half the number of chromosomes. They're already in what's called a haploid number, and that means half the chromosomes. Let's see if that was written here somewhere for you. Half the number of chromosomes. It's not written, but it's called haploid. Yeah, it is. Haploid. Okay? So after the first division, they're already in. It's 2 cells. 1 cell divided into 2. They're already in a haploid situation. So why did these 2 need to divide? Well, the reason is because the chromosome is still in its duplicate form. So the partners lost their partnered pair of the, and remember, it's not just 2 pairs, it's 23. They're just showing you because this would be so confusing seeing all 23 of them lined out. But now they have to lose their copied pair, their sister chromatids. So what will happen in the second division is that these two cells will undergo another division. And when these 2 cells undergo another division, now you have 4 cells. And if you just look at this illustration, I know it's just an illustration, but it's accurate. If you look at this sperm compared to this sperm, compared to this sperm, compared to this one, we started with 1 cell. It went to 2, and then the 2 divided again, and now we have 4. These 4 cells, none of them are alike. If you look at this chromosome, that was the homologue of this one. It's different than that one, different than that one. This homologue is different from this one, different from this one, different from this one. I want you all to think about that. I want you to think about males' sperm, making sperm. And now I want you to think about something else. I want you to think about these little sperm that are made, and then they're maturing, they're getting a little tail, they've got a little head here. And I want you to realize that males, in a teaspoon has 5 mLs. So everybody can picture what a teaspoon of, I don't know, water looks like. So think about a teaspoon of semen. So in 5 mLs in a teaspoon, for every 1 ml, so 1/5 of that teaspoon can have 50 to 120 million sperm. So now you're thinking about a teaspoon. 5 times the number. So 1 cc per 1 cc is the same thing as an ml. So 1 ml normal should have 50 to 120 million sperm. That's normal count. And of those that are analyzed, there needs to be a majority percentage need to be active and need to look anatomically like this, or let me get to one. Look anatomically like this mature sperm. Sometimes when you do a semen analysis, you'll see a certain number of sperm that are in that right range of number, but they may not have a tail, or some of them may not have a tail, or some of them may have two tails, or some of them aren't moving. So that all gets evaluated on a semen analysis. But here's what I really want you to think about for a teaspoon of semen. And I want you to think about males starting at puberty, how often, let's say in a week, maybe even a day, ejaculations happen, right? For their lives, if they live to be, let's say they live to be 80, okay? How many ejaculatory events happen? And think about in 1/5 of a teaspoon, there can be 50 to 120 ml. That's the normal amount of sperm. And I want to let you know something. No two sperm a male will produce in his entire lifetime, no two are identical genetically. The chromosomes. There is such, because of meiosis, which is such an amazing thing. Meiosis, this is why you should be impressed with meiosis. The male starts, the first cell has the male's 46 chromosomes. They all start out the same. But when that one cell undergoes meiosis, 2 divisions, it shuffles, these homologous partners get together and switch off genes and shuffles these chromosomes so that a male in his normal lifetime, starting from puberty until death, with all of the sperm that they make on a 24-hour cycle, will never have 2 sperm that were genetically identical. That's just phenomenal. That's truly phenomenal. You might want to listen to that again, because that could blow your mind, really, if you were listening to that. It's going to be the same way in women, but females don't make nearly the number. We have hundreds of thousands of primordial eggs, but during our reproductive capable years, from puberty until menopause, where we stop producing eggs, we produce 1 usually. Sometimes women produce 2, and then they have chances of twins, right? But typically 1 a month. So in women, it's not about the quantity, and it's truly about the quality, and it's also about the quality of sperm in males too. It definitely diet, lifestyle, all that plays a role in how well sperm are being, made. So I'm not saying it's not about the quality of sperm also for males, but males have a lot of quantity of sperm. They're making a lot of sperm. If you think about, though, a female only has to be near a reproductively capable male. I'm thinking of this like animals, because we are. I didn't say we have to act like it, but we are. Really just for a short window of time, once a month. But males have to be ready every day. They have to be kind of ready every day, because they may be encountering a female that's ready. So that's the way that it is. All right. So we're looking at, you can go through some of these, but I talked about the blood testes barrier and how important that barrier is to protect newly developing sperm that are being released into those little ducts, those little testes, the little ductwork inside the testes, because you do not want them being exposed to the male's immune system. Because remember, the sperm has the first cells have a diploid number, have a 46 number of chromosomes, but once these sperm are made, these are foreign, really, to the male. So anyway, you can look at some more interesting things about sperm if you want to. Packed into the head of the sperm are the chromosomes, because really the sperm just has a job to get to that egg and then be able to bore into that egg. The end of this head has a what's called acrosome. This has digestive enzymes in it. So anytime the sperm butts into anything, digestive enzymes are being released from the acrosome. So once it gets to an egg and it's bumping into that egg, it's trying to digest through a zone of the egg that's protecting the egg, and the first one that gets through is the sperm that, contributed to fertilization. So this acrosome contains digestive enzymes. The head is holding back the chromosomes that are going to be delivered to the egg that has the other half of the chromosomes for that zygote. And then really the rest of this has to do with moving this cell, which is going to be the tail, the flagellum. This is called a flagellum. And there's mitochondria here because it's going to take a lot of energy to move it. These sperm can live for a few days, and need to. So, and then they would die. So this is what I was telling you about the fluid expelled. It's not always, sometimes that can be a problem if it's not always at the same time. But anyway, I'll talk about normal. A lot of it is seminal vesicle, so you would go back and see where that seminal vesicle is. About 30% is the prostate. About 10% is the sperm, and so the bulbourethral gland also contributes. The function is going to be providing energy, helping with protecting the sperm against the vaginal secretions. Vaginal secretions are really, really kind of not friendly to sperm. Vaginal secretions are trying to protect a female, but the sperm have to protect themselves from that. And so, anyway, these are just all some of what the functions of semen is. And here you can see that per 1 ml, a normal count is 50 to 120 million. And again, never in a lifetime do men have 2 identical sperm genetically. Okay, so I don't really ask any questions about sexual excitement. I feel like this is like a separate class, but a climax or orgasm and the reaction that occurs discharges the semen ejaculation. Sometimes ejaculation and orgasm are not happening at the same time. That can be a problem sometimes. But anyway, that's just the terminology here, and I think that that's understandable. Another difference between males and females is that, typically after ejaculation there is a period called like a resolution period where, it might be minutes, it might be a couple of minutes or a few minutes for some people much longer, where another erection, it's going to take time for another erection or orgasm. So I guess that's individual. But anyway, so this is that, and I don't really, I don't ask any questions about that really, other than understanding the terminology and what an ejaculation is versus an orgasm. But anyway, now this is the end of the male reproductive. So we will go into the female reproductive, and we will, at the end of the female reproductive, I will talk about sexually transmitted types of diseases that of course affect both males and females, not just females. And what we understand about many of those is that quite frequently, often actually, more often than not, they are asymptomatic. So that means that there are no real signs or symptoms with them. So this is why people who, once they become sexually active, whether they are monogamous or not, they should get routine screenings, because many of these particular sexually transmitted diseases, as I said, are asymptomatic, so you would not know unless you got screened. And you would want to know because some of them are completely, you would want to know for a lot of reasons, but some of the reasons is that you would want to know because many of them are treatable and curable, like completely curable, but you would want to also treat partner or partners or whatever you would want to be treated. Some are sometimes some of them are not treatable, so it's things that would be latent that people would have. But again, we're going to talk about some of those. Some of them can affect fertility because, as we said, with all the duct work that we think about with reproduction, it's not just about testes and ovaries. The sperm have to actually meet the egg, and that's a long journey. It's really a long journey. So all this duct work in the male, and we're going to look at the duct work in the female. Those ducts have to remain healthy, and those structures have to remain healthy. So if there have been infections that can actually lead to scarring of the tubules, or then this could end up potentially causing fertility issues. So another reason to catch things really early, get them treated, so that there's less chance of that actually happening. There are some sexually transmitted diseases that actually can become life-threatening diseases, ones that can end up leading to pelvic inflammatory diseases of a female, and then obviously we know about HIV, but we'll talk about these some more in the female. This is all for the male reproductive system. So I hope I haven't forgotten anything. If I have, I'll try to add it to the female discussion. But anyway, see you with the female reproductive system the next lecture.